Hi, hi everyone. We're the Madi Lab, MP003, and our project is designing and developing a recombinant mRNA vaccine against the Nipah virus by targeting fusion protein binding between mammalian and viral glycoproteins. I'm Caleb.
I'm Raj.
Anishka. I'm Shreya.
And I'm Aaron.
Okay, so just for our table of contents and what we're going to discuss today, so we're going to start with background information and then hypothesis, and then our experimental design, our methodology, our results, and then our conclusion.
Okay, so to start us off with the background information, the Nipah virus is a zoonotic pathogen that has been known to lead to death in 40 to 75% of human cases. It is a biosafety level 4 pathogen and causes a high risk in public health and security since there are no effective FDA-approved vaccines. The Nipah virus first emerged in Malaysia and spread through Asia to places like India and Bangladesh. Cellular interactions by the Nipah virus are controlled by the fusion and attachment proteins, or F and G. EphrinB2 and Ephrin B3 are the receptors that the G glycoprotein binds to. Eventually, the inner membranes of the F protein and the host cell allow for viral entry. The antibody 5B3 has been shown to have a high binding affinity to the Nipah virus glycoprotein F, which illuminates the importance of interactions between the antibody and the F protein. This is the main key to our development of the mRNA vaccine library.
Our research question for this project was, can we develop a potential vaccine as a blueprint against the Nipah virus in the form of mRNA? So an mRNA vaccine. And our null hypothesis was that the synthesized mRNA vaccine and variant library of the F fusion and G attachment glycoproteins does not elicit a productive immune response. Our alternative hypothesis was that there is a significant immune response to the synthesized mRNA vaccine and vaccine library variants. Our experimental design has 4 main steps that we will repeat. First, we generate the Nipah virus plasmid. Then we add the T7 promoter to the plasmid and generate mRNA from that. Lastly, we'll check for an immune response from our CHO cells, and then repeat this entire process for each plasmid generated by a vaccine library, which will be discussed on the next slide.
So as mentioned, our goal is to generate an mRNA vaccine library that encompasses several variants of the Nipah virus's proteins F and G, and that will theoretically attain higher transcription rates, avoid detection by the host's immune system, and better infect the host. To create our library in silico, we have identified binding residues that are of most importance based on literature and PDB-PISA. Then we generated multiple mutated residue sequences, and we conducted point mutagenesis on PyMol, and we substituted these fragments into our proteins and ran them on a docking server, ClusPro. Then we compiled a list of sequences that yielded the largest negative Gibbs free energy values. As we create these variants, we are also in the process of synthesizing our F and G proteins in vitro. First, we developed the plasmid of each with ampicillin resistance and our protein of interest. Next, we transformed our plasmid into E. coli, which basically means like mass production of the DNA. We then extracted the DNA using mini-prep, and we performed a restriction enzyme digest to linearize the plasmid. After DNA ligation to add the T7 promoter, we sent off our work for sequencing to validate our linearization. So as we confirm our linearization, the next steps will include proceeding with transcription of our DNA into mRNA, which is a precursor to proteins of these variants. And once we get a final list of these variants, we will repeat this process and generate mRNA to conduct our immunogenicity study.
Okay, so for our computational results using PDB-PISA, we identified the largest negative delta G among residues in F to be asparagine 464. Then using ClusPro, we compiled a list of weighted scores of the docking of the F protein and an antibody we found in literature point mutating asparagine 464 with every single amino acid through PyMol. We then found that when substituted with leucine, the F protein results in the highest binding score. However, the difference in binding scores were not very significant, as you can see here in the graph in the middle. As for our lab results, we have gone through with our plasmid transformations, extractions, enzyme digest, and annealment of our T7 promoter sequences. However, after sending it in for sequencing, we found that we actually had two sites where ECAR1 has cut, as seen in the gel on the right here, which essentially cut out our T7 sequence, rendering our gene of interest unable to transcript.
Okay, so for conclusions for mRNA synthesis, since like Caleb said, the execution of DNA
打
ligation
放在
to
锅子
insert
里面，
the
很
sequence
漂亮。
into the plasmid
那只2个，我想说明天快点。
wasn't successful. We're gonna revisit the ligation process with a new restriction enzyme called SAC1, which we confirmed has only one cutting site, so our linearization will be successful. And then we'll also fine-tune parameters such as vector and DNA insert concentration, as well as the reaction temperature to increase the to increase its success. Future work for the mRNA synthesis would be that once transcription is completed to RNA, we will add additional post-transcriptional modifications such as a poly-A tail and a 5' methyl cap to improve stability. And we will encapsulate our mRNA in a lipid nanoparticle and transfect into CHO cells to test for the vaccine efficacy and a productive immune response. And for our vaccine library, we currently have located residues that are essential in the fusion process through literature research and analysis of the binding cavity of the fusion protein via Gibbs free energy values. And basically want our vaccine library to mimic an environment in which the Nipah virus naturally mutates and adapts while increasing its survivability and reproducibility. So for next steps, would be for the mRNA vaccine library development to include performing gene shuffling via SwissPro, which would increase the diversity of the sequences. And since substituting our point mutated sequences was insignificant, as Caleb said, we will be shuffling non-conserved regions and evaluating their binding efficiency to the 5B3 antibody in autodoc vena. And once we do that, the sequences that produce a negative vena score will be biologically relevant regarding their immune response generation and will be inserted into our library. We would like to thank Mr. Amati for giving us his guidance on this project and ASGRP for giving us the access to the lab and resources and staff whose insights and help have been invaluable. Thank you.